Hello everyone and welcome to today's presentation on testosterone-induced skeletal muscle hypertrophy. My name is Samir.
And my name is Kush.
Resident muscle stem cells, also called satellite cells, are adult stem cells located in skeletal muscle cells. These stem cells become active in response to muscle injury or exercise and give rise to skeletal muscle cells or myofibers. The term myonuclear accretion is used often in this paper to describe the growth of muscle cells using satellite cells. This figure is showing how an injury to a myofiber results in the regeneration of a new myofiber through the activation of satellite cells. Next, we're going to look at the importance of satellite cells in muscle hypertrophy. Hypertrophy is a term used to describe muscle growth. As we said, satellite cells are muscle stem cells that give rise to myofibers. Previous research has shown the presence of satellite cells are necessary for postnatal skeletal muscle growth and regeneration, but their required presence during times of mature skeletal muscle hypertrophy is currently unknown. There have been studies that have focused on the requirement for satellite cells to be present during muscle growth in adolescent and mature mice, which have shown that the presence of satellite cells is necessary for muscle growth before complete maturation. It has been found that when mature muscle cells sustain long periods of muscle growth, satellite cells are required. This figure on the bottom left shows how satellite cells can have different functions, playing roles in homeostasis, inducing muscle growth, and regenerating more satellite cells. The figure on the right shows where satellite cells are located in relation to myofibers. Now we're going to look at how testosterone treatment stimulates muscle hypertrophy. Testosterone is an androgen that has consistently been shown to promote the number of satellite cells, an increase in myonuclear amount, and an increase in muscle fiber cross-sectional area. With an increased number of satellite cells, testosterone will stimulate muscle hypertrophy. Testosterone binds to specific receptors known as androgen receptors, which release from the membrane to stimulate protein synthesis, phosphorylation of specific genes, and insulin growth factor 1 expression. It has been found that testosterone treatment of satellite cells in vitro results in a 25% increase in proliferation. Even though we know that administrating testosterone results in hypertrophy, the direct relationship between an increase in satellite cell content, testosterone, and hypertrophy is not known. Next, we'll discuss the PAX7 DTA mouse model. PAX7 is a transcription factor found in mice that is important for muscle tissue growth and regeneration, or myogenesis. The PAX7 mouse strain can be crossed with a ROSA26 mouse strain in order to obtain the PAX7 DTA mouse model, which is useful in enabling the depletion of satellite cells. There are two types of PAX7 DTA mice used in this research. SC+ are mice with satellite cell replete, and SC- are mice with satellite cell deplete. Within the SC- mice, tamoxifen is induced. Tamoxifen is a drug that can be used to deplete PAX7 DTA mice of satellite cells. This can further help us understand the relationship between increased satellite cell content, hypertrophy, and testosterone. The synergist ablation model was used in this research. This method highlights the sole efficiency of the muscle of interest by surgically removing surrounding muscles. In this research, the muscles of interest are the soleus, plantaris, and extensor digitorum longus. By surgically removing the surrounding muscles, it puts more tension on the muscle of interest, giving it the ability to grow at a faster rate since it is working alone. This figure is a representation of the synergist ablation model. The figure shows how the removal of the gastrocnemius and soleus muscles helps in isolating the plantaris muscle to not allow it to work in synergy. The hypothesis of this research is that testosterone-induced muscle hypertrophy is driven directly by satellite cell activation and subsequent myonuclear accretion.
So let's dive into the results of this research. First, the authors wanted to test if testosterone administration induces a higher whole body weight in mice. So there are 4 different experimental groups, which are categorized under 2 sections. The first section is sham, and the second one is tested. Sham is also known as the control group, whereas tested is also known as the experimental group, where testosterone is given. The results of the experiment are cataloged in a graph where the x-axis is our period of time over 3 weeks and the y-axis is the body weight of the rat muscles. The results reveal that the addition of testosterone causes an overall increase in muscle mass and body weight in tested SC plus and SC minus rats. The researchers are now looking at 3 muscles in rats, soleus, plantaris, and extensor digitorum longus, and they're seeing the individual muscle weight. On the x-axis, you can see our control group, sham, and our experimental group tested, as well as SC plus and SC minus for each group. And on the y-axis, you can see the muscle weight for each of the 3 different muscles being studied. Now, the authors are seeing if testosterone affects the muscle weight in each of these groups. The results show that with or without satellite cells in rats, testosterone seems to increase the muscle mass. However, when satellite cells are present, it can induce a greater increase in muscle growth. This can be said when we compare SC plus tested and SC plus sham to SC minus tested and SC minus sham. Between SC minus tested and SC minus sham, there is only a small increase in muscle growth. The authors now wanted to see the cross-section of the plantaris muscle and observe the effects on cells during testosterone treatment in satellite cell-depleted rats. Through immunohistochemistry, the authors are visualizing the muscle fiber size. When testosterone is added, the size of cells in testosterone SC minus increases drastically in comparison to sham treatment. This indicates that testosterone can increase the size of myofibers without the presence of satellite cells. Next, the researchers are now quantifying the data of how much the muscle fiber grew when testosterone was added in each of the 3 muscles. From these graphs, you can see that on the x-axis we have our sham and testosterone groups. Within each, there are SC plus and SC minus mice. And on the y-axis, you can see the mean fiber cross-sectional area per micrometer squared. And so from these graphs, it can be observed that the SC minus mice exhibited the same hypertrophic response to testosterone as SC plus mice, thus revealing that there may be other factors that can help increase muscle fiber CSA besides satellite cells. So the researchers now wanted to observe the phenomenon of myonuclear accretion. Myonuclear accretion represents the repair and regrowth of muscle tissue after undergoing stress. The authors wanted to see myonuclear accretion when testosterone was administered in the presence and in the absence of satellite cells. Here, the authors used immunohistochemistry to stain the myonuclear fibers. From the results, it can be seen that when satellite cells are present along with testosterone, more myonuclear accretion seems to occur due to the increasing number of myonuclei. Lastly, the researchers wanted to quantify the data from the immunohistochemistry. They look at soleus, plantaris, and extensor digitorum longus muscles. On the x-axis, you can see the 2 groups of sham and testosterone, which contain both SC plus and SC minus mice. On the y-axis, you see the amount of myonuclear fibers. The amount of myonuclear density was greatest in soleus, followed by plantaris, and lastly in the extensor digitorum longus. Without any satellite cells, there is no myonuclear accretion. However, in comparison to previous data, we can observe that SC minus mice do exhibit similar hypertrophic response to testosterone as the SC plus mice. So let's revisit the hypothesis. Testosterone-induced muscle hypertrophy is driven directly by satellite cell activation and subsequent myonuclear accretion. From these experiments, it can be observed that the fusion of satellite cells may be necessary for myonuclear growth and later on skeletal muscle hypertrophy. However, in these previous experiments, it can be also observed that skeletal muscle hypertrophy can be achieved without satellite cell fusion.